Assalamualaikum and Hey to Prof. Shah and fellow classmates. We are from Group 1. We'll present about the foodborne pathogens for Applied Food Microbiology course BMY 4309. This is the list name for our group members. For your information, we are dividing our group members into two teams. The first team will do the slides and the second team will do the presentation. Okay, what is foodborne pathogen? Foodborne pathogen, according to Binti's 2017, is the microorganism that leads to foodborne illness upon the consumption of the contaminated food due to the production of toxin. For example, the foodborne illness is like food poisoning and also diarrhea. So, for your information, the rate of foodborne illness in Malaysia is quite high. As you can see from the picture, the dirty environment is one of the major factors which cause the foodborne illness. The dirty environment will promote the bacterial growth. This will lead to the food poisoning cases. For your information, recently there is food poisoning cases in Terengganu, right, which involve the pudding buih. It is quite severe because it caused few deaths because of the pudding buih. I think the causes of the illness is because of the contamination of bacteria in the pudding buih. So 50% of cases of food poisoning cases are due to unsanitary food handling. As you can see from the picture, some restaurant, especially Mama restaurant, has this environment at their kitchen. This is very worrying because this dirty environment will promote the growth of pathogen that will cause many foodborne illness. One of it is food poisoning. So this is the example of pathogen that cause foodborne illness, such as Clostridium perfringens, Campylobacter species, Salmonella species, Staphylococcus aureus, E. coli O157H7, Listeria monocytogenes, norovirus, and Toxoplasma gondii. So for your example, E. coli O157H7 was first identified as a pathogen in 1982 in an outbreak of bloody diarrhea traced to hamburgers from a fast food chain. So this pathogen is originally a food-borne pathogen. So what is new foodborne pathogen? The new foodborne pathogen is a known microbe that causes foodborne illness where previously its transmission was not through food. So for example, Campylobacter jejuni, it is known as a rare opportunistic bloodstream infection until veterinary diagnostic methods used on specimens from humans showed that it was a common cause of diarrheal illness. So, which means originally it is not transmitted through food, but now it is it is known as one of foodborne pathogen. So, what is the factor that lead to the discovery of new foodborne pathogen? The first one is human demographic related. Some of the example is the change in eating habits and consumer preferences. The people nowadays prefer to eat ready-to-eat meals, fast food, raw food, and also takeaway food. This will lead to the growth of foodborne pathogen. The second one is the lack of exposure on the importance of food hygiene. It is important to practice a very good hygiene in food handling, preparation, and storage of food. And the third one is the increases of international travel. The increases of international travel can cause the changes of microbial community from one country to another country, which means the travelers will bring back the bacteria which are not origin in our country. So for the next factor, I will pass to my group mate to present.
Thank you, Amalina. In addition to Amalina's point on the factors that causes the re-emergence or the emergence of foodborne pathogen is food-related. The factor food-related basically tells us about the environment the food is in. So changes in food production and distributions, changes in food production, processing and packaging. So the difference between these two is in the distribution, it's more related to the wet food, where large facilities are allocated for animal slaughtering and then they are transported to wet markets or supermarkets or markets to be sold. While in processing and packaging, it's more related to the dry food itself. For example, canned food, sardined canned food. And in processing, the manufacturer will tend to reduce costs by lowering the salt concentration or reducing the heat treatment itself. So this is one of the factors why the microbes can still survive despite being killed by despite undergoing heat treatment itself. And of course, packaging, the material of the packaging, whereby the material definitely has to be a solid one. And if we see, for example, for most of the canned food, it's in a solid zinc covered, it's solidly covered with a metal base. So if it's supposed to be a loose, a weak one, definitely it can easily spoil the food can easily spoil because they can be easily dented where microbes can just enter. And next is global marketing, whereby hygienic procedures are not being thoroughly followed. Moving on to the next factor is pathogen related. So in pathogenicity, we can actually observe from the current pandemic itself, where the SARS-CoV-2 is a virus which mutated and adapted to the current environmental changes, where it can survive the heat, it can survive the cold, and now it has high rates of infection among humans. So just like that, even foodborne pathogens can undergo mutation and adaptation to stressed environments. And the next subpoint would be advances in identification of pathogen. For example, molecular identification. And another point which is not stated here is actually the ability of the pathogen to resist antibiotics. It has a high antivirulence factor where even antibiotics don't work on them. And the next factor, agriculture and environmental related, where we can see increase in large farms with increased animal production is also a factor to be considered. Because just like in the 1990s, to be specific 1998, the outbreak of Nipah virus in a pig farm in Negeri Sembilan. So even the farmers were infected with the virus. So in an animal manure, virus, although it's a virus, but it can be related to foodborne pathogens as well, mostly with bacteria. So basically, when there's a big farm, there's chances that the microbes can spread more than it has already spread. And next is the ability to sustain climate and ecosystem changes due to the changing environment now. And another point is the antibiotics, previously mentioned from the pathogen-related factor, where antibiotics are mostly being used to feed the animals, so that the animals will be having a high resistance to diseases, but microbes can overcome that in a particular uh, uh, in a sense that they actually can develop the resistance towards all these antibiotics. And lastly, the last factor on the re-emergence of foodborne pathogen, is because of the public health and the social life itself. So, inadequate facilities and programs, where some are really adamant on not having a vaccination, so due to some controversies, and so when this happens, we lose the herd immunity in there. So the susceptibility towards food pathogens become even higher. And lastly, social trust. For example, civil wars and bioterrorism.
Next, the mutation toward foodborne pathogen. So the first one is Listeria monocytogenes. So this is a gram-positive bacteria that cause listeriosis, which has 20 to 30% mortality rate. So this Listeria monocytogen have internally genes, which is INLA and INLB, that help in the penetration across the internal barrier. The mutation in Listeria monocytogenes is the mutation of INLA genes. So the mutation of this gene result to premature or stop codon, and this mutation can happen either by the deletion of adenine or the frameshift mutation, creating a nonsense codon. So next, this mutation in Listeria monocytogenes is due to the cross-contamination and also unhygienic food retail environment. So cross-contamination can occur when uncooked food, such as fresh raw vegetable, is combined with cooked meat, cooked food such as a cooked meat. So when the uncooked fresh raw vegetable is combined with the cooked meat, the cross contamination between the fresh raw vegetable towards the cooked meat. Then the unhygienic food retail environment, as we know, it is very common in Malaysia to open a food stall beside the road, and this sometimes beside the road is an unhygienic environment toward the food. For example, maybe the food stall is beside a drain or in an area with a lot of pests going around, such as rat, and also a polluted area. So this unhygienic food food retail environment can cause the mutation in the Listeria monocytogenes. So other than that, the person that handle the food, or the tool that are used to cut, maybe the knife that used to cut the food, or maybe the surface itself that they used to put the food, is contaminated with other microorganism and also Listeria monocytogenes. So the impact of the mutation, the first and the one that we fear the most is the resistance to the disinfectant. Due to the mutation, the Listeria monocytogen is resistant to disinfectant such as ammonia compound sanitizer. Because of this, scientists or researchers need to find a new disinfectant or a new way to kill or prevent this bacteria from contaminated the food. Next is the enhance of pathogenesis of bacteria. There's genes that emerge due to this mutation, IISX genes, or its other name is LIPI3, which stand for Listeria pathogenicity island 3. So this gene is a very important virulent factor for this bacteria because it enhances the hemolytic and the cytotoxic activity of the Listeria monocytogenes. So because of the mutation, the bacteria such as Listeria monocytogenes become more pathogenetic and also become more aggressive and virulent. So the next one is Staphylococcus aureus. So this is also a gram-positive bacteria that cause staphylococcal enterotoxin. So the enterotoxin that secreted by the Staphylococcus aureus can cause a toxic shock-like syndrome, allergic, food poisoning, and also other autoimmune disease. So the mutation in the Staphylococcus aureus can cause the emergence of methicillin-resistant Staphylococcus aureus, or in short, MRSA. So the MECA genes encoded for the penicillin binding protein, PBP2A, has a low affinity to the beta-lactam, and this will cause a resistance to beta-lactam. So as we know, beta-lactam is the one of the mechanism that is used by the antibiotics such as penicillin to penetrate the cell wall and kill the bacteria. So because of the resistance to the beta-lactam, so these Staphylococcus aureus become resistant to that antibiotic. So the emergence of methicillin-resistant Staphylococcus aureus can either by gene transfer, such as plasmid and transposon. Transposon is other name is called jump gene, where the genes can jump from one strand of DNA to another strand of DNA. And also emergence of MRSA can either happen by the insertion mutation or deletion mutation.
Assalamualaikum and greetings. I'm going to continue the mutation in foodborne pathogen. Mutation in L monocytogen is because of growth promoter and antibiotic residue in food, because usually in animal production, the company always use antibiotic, so that can cause mutation in L monocytogen. Okay, next is Escherichia coli. Escherichia coli or E. coli is a gram-negative facultative anaerobic and commonly found in the lower intestine. And E. coli can cause diarrhea, pneumonia, and urinary tract infection. Sometimes it also can cause vomit and acute kidney failure. E. coli produces a toxin that is called sugar toxin, which can damage the lining of teeth. Mutation in E. coli is usually involved of lactose plus. The mutation of lactose plus is the spectrum of adaptive lactose mutation. This is involved of friendship mutation, and that is recombinant dependent mutation. In recombinant dependent mutation, let A, the repressor of the S regulon in E. coli, repress both rate A and group AB operon, and that is group AB and rate CG in that plus active mutation. In this mutation, the translocation toward the fork pairing of two newly synthesized strands of DNA. The newly synthesized DNA is not immediately methylated, and the transition state of polymethylation allow the mismatched repair system to correct a mismatch in favor of the methylated template strand. And another one is DNA polymerase involved in plus additive mutation. This is all the mutation in E. coli. The next mutation in E. coli is because of gamma and UV radiation, recombinant dependent mechanism that involve the genome and protein and subpopulation of cells that have a transient mutator phenotype. And the next pathogen is Clostridium perfringens. This bacteria is a gram-positive, root-shaped, anaerobic, spore-forming, and spore-forming. This bacteria lacks flagella, so not easily moved, but still migrate across the space using a type of gliding motility that involves the formation of filaments. Clostridium perfringens can cause food poisoning, gas gangrene, tissue necrosis, and emphysematous cholecystitis. Okay, mutation in Clostridium perfringens it involves mutation in translation. As we know that translation is the step or process from RNA, the RNA that step after the transcription, right? The RNA that been translated to produce amino acid or protein. Substitution mutation in Clostridium perfringens involves substitution mutation. As we can see here, from GAC to GAA, which is aspartate. Aspartate produce aspartate, but produce glutamate from CTT to GAT, which is alanine to aspartate, from GTT to TGG, which is glycine to tryptophan. And another mutation also involved is translation mutation. This also involves in translation also from GAG to TAG, from glutamate to stopprodone. It usually involved in the RNA or RNA that is used to translate to protein and amino acid. So because of the mutation, the protein produced, the amino acid and protein produced also will be different. So the next is the impact of the mutation is complementation of cell division mutation. This is which is led to a reduction in the average of cell length, a longer lag time for strand carrying plasmid, and exhibited a higher level of cell lysis after it reached stationary phase because the protein is produced, right? So it's another. So the function will also differ and causing a unwanted effect. So that's all from me. I will pass. I will pass to my friend to explain subtopic subtopic for foodborne pathogen. Thank you.
Okay, so Assalamualaikum and very good day. Now I would like to continue the presentation on strategy in controlling the food pathogens. So first, they are divided into two parts. One is the control of source, another one is the control of transmission. Now I would like to focus more on the control of source, and actually there are three control of source, which are the closing of food premise first. So the closing of food premise will be done by using the agreement of the business, or it might be enforced by the law. And second is, it should be remain closed until the appropriate authorities approve their reopening. Means they really cannot open it illegally, and also really need to wait for the proper authorities so that only they can reopen back. And third is the reopening of establishments may vary by the jurisdiction. This jurisdiction means that is the to make the judgment or the decision by the legal power. Okay, so now moving on to the second point, which is the removal of implicated food. Actually, these fresh products generally consumed within 3 to 1 week of the production and make the likelihood recovery is poor. So the frozen food or shelf stable, like for example, like the canned foods, packaged foods, that kind of products are most likely to be recovered as there is a less urgency to move them through the system. Okay, after that, under the implicated of the food, the removal of the implicated food, there are 3 points, which are the procedure, communications with the public, and post-recall reposting by the business. So focusing on the procedure is actually once the appropriate authorities have decided to recall the food products, they should communicate with each other and ensure the cooperation of the business involved in the recall. So actually, first of all, the food recall is meant. The meaning of the food recall is the action taken to remove from the sale, distribution, and consumption food which may pose a safety risk to consumers, and may be initiated by the results of a report of complaint from the variety of sources. For example, like the manufacturer, wholesalers, retailers, government agency, and the consumers as well. So they should, and then still on the procedure, they should directly advise the local health authorities of the recall and any enforcement actions required. And they also should ensure the appropriate public notification. So moving on to the second, which is the communication with the public, is actually the information the public should be included is actions of the consumer should take if illness occurred, and appropriate food handling information for the consumer, for example, like name and brand of the food products, including the labeling, and then the associated, a description of the common symptoms of the illness associated with the suspect pathogens or the contaminant. So moving on to third, which is the post-recall reporting by the business. Business should provide the food safety committee or other appropriate authorities with interim and final reports, and the recall, which should contain the following information, which are result of the recall, method of disposal or reprocessing of the recovered stock, and difficulties experienced during the recall, the action that proposed for the future to prevent any recurrence. So moving on to the final one, which is the modifying food product process. The corrective action must be taken immediately to avoid any recurrences. For example, the modification of the recipe or of the process means you can try something new recipe or any new form of cooking process, actually you're trying to modify it. And second is a reorganization of working practices, means you need to reorganize like everything, and change in storage temperature means you should apply the proper or optimal temperature which is suitable for that specific and particular food products. And then modification of the instruction to consumers means you should give the proper instruction uh, to the consumers, and that's all. So now the presentation will be continued by the next presenter, and thank you.
Thank you, Farini. Hi, I'm Maharitya. My matrix number is 196375. Here I'm going to talk on another strategy in controlling new food pathogen, which is by controlling the transmission of the food pathogen. Let's get into it. Okay, there are three types of controls that we have come across, such as public advisors, isolating or making people quarantine, and protecting risk group. Okay, public advisors can be given for especially food handlers to maintain their way of handling food, such as asking them to wear gloves when handling the food in order to prevent the contamination from the hand to the food, or asking them to not use the same equipment that they use to prepare food. Another advice can be given for maintaining the personal hygiene measures, such as after leaving the washroom, it is a must to wash hands in order to not contaminating the food source with the fecal contaminants. Yeah. Another control is isolating infected person from public area. This one is to prevent the infected people from contaminating the food source. Specific advice and protection for risk groups may be considered in some circumstances. As example, the groups of pregnant women or immunocompromised persons, such as those having human immunodeficiency virus, or when the HIV has developed into AIDS, should not consume unpasteurized milk or cheeses, raw fish, and other foods that potentially contaminated with Listeria monocytogenesis or Listeria species. Another example is persons with or people with liver disease should not consume raw oysters that may transmit the Vibrio bacteria. And yeah, that's how we can protect the risk group.
For the last part is current issues of emerging foodborne disease and their solution. For the first issue, which is salmonella infections in eggs. In June 2019, there were 17 reported clusters of food poisoning outbreaks related to one restaurant in Hong Kong, affecting 40 people. This spoilage caused by preparation of eggs from unpasteurized eggs 1 day beforehand and stored in refrigerator at 9 degrees Celsius. Eggs were inadequately heated to a semi-cooked condition on the next day and kept in lukewarm water bath at 30 degrees Celsius. There were no further reheating or cooking of the eggs was done prior serving to customers. The solution for this issue is thoroughly cook egg dishes and prepare scrambled eggs on a per order basis. Use full eggs as soon as possible and avoid topping up. For preparation of dishes without heat treatment, the advice is to use pasteurized eggs, as pasteurization heats eggs to a point that kill any dangerous bacteria but do not cook the egg itself. The second issue is listeriosis. A dangerous outbreak of listeriosis bubbled out of Michigan late February involving enoki mushrooms grown in South Korea. Case reported. In April 18, 36 confirmed cases in 17 states. For the 18th April, 6 people in Australia were identified as part of an international Listeria outbreak traced to the imported mushrooms. The solution for this issue is washing and sanitizing all food surface and containers is important for anyone who handle enoki mushrooms or any other food contaminated with Listeria. CDC advises washing surface with hot soapy water and washing containers in a dishwasher with hot soapy water. Food processors, restaurants, and retailers who handle enoki mushrooms need to be extra vigilant in cleaning and sanitizing to avoid cross-contamination. The third issue is E. coli 0103 clover sprouts. The firstborn disease outbreak reported in 2020 is caused by E. coli 0103 associated with products containing clover sprouts by Chicago Indoor Garden. Cases reported on March 19, the outbreak involved the rare E. coli 0103 had sickened 39 people in 6 states in the US. Few samples were collected and analyzed. The presence of E. coli 0103 were identified. Whole genome sequencing of this bacteria shown that it matches the current outbreak strain. The solution for this issue is Chicago Indoor Garden has recalled all their products containing red clover sprouts. Cook sprouts truly to reduce the risk of illness. Truly, cooking sprouts kill the harmful bacteria. Children, older adults, pregnant women, and people whose immune systems are weakened were not advised to consume any sprouts for the time being. Generally, outbreak of foodborne illness can be prevented efficiently by adequate hygiene in storage and food handling, such as store food in specific container in suitable temperature, avoid keeping food for a prolonged period, don't mix food with raw food or use the same utensils to prepare them. Wash food thoroughly before serving or cooking. Cook foods to its minimum required temperatures to avoid bacterial growth. Keep cold food cold and hot food hot. For the conclusions, foodborne disease will continue to be a matter of major concern around the world in this foreseeable future. Despite some important national success at reducing the levels of certain pathogens in food, public education is seen as a key factor in improving food safety practice in the home. The benefits of food hygiene education, such as food handling and processing, in maintaining the national economies. That's all from our group. Thank you.